Hello, everybody. Welcome to the meeting. I'm here to introduce Antonio Giraldez, who's going to be our wonderful first keynote speaker this evening. So Antonio did his undergraduate studies in Spain, and then he went and did his PhD at the MBL, working with Steve Cohen, studying notch signaling and wing development in Drosophila. After that, he ended up in Boston and worked with Alex Shear, where he began to study zebrafish and really began to develop an understanding of a role for microRNAs during the embryonic transition, and in particular microRNA, miR-430, that plays a huge role during this developmental process. So since 2006, Antonio has been at Yale, and he ran a graduate program for 4 years and got a taste for leadership, and he has now assumed the role of Department of Genetics chair, and he says he's pretty busy. But we're really happy he could come and speak. He continues there to work on the molecular mechanisms of gene regulation and how they shape embryonic development and post-fertilization processes. And really, these questions are at the heart of the meeting. I think he's going to give us a wonderful talk on a variety of approaches that he's really leveraged to give us an understanding of gene regulation during these early stages. So please join me in welcoming Antonio for our first talk.
Right, thank you so much, Tom and Davide and Rachel, for the kind introduction. It's really what I think is an undeserved honor to be here and opening the meeting, so I'm very humbled by this invitation, and I'm really happy to get your feedback in the project. And particularly, this is all the work of extremely talented, wonderful colleague, Jean-Denis Boudin, who's actually a superb RNA biologist and is in the job market this year. So he's at the meeting, he's presenting Post-it 30, and I really feel bad that he's not actually giving this talk instead of me. And the punchline of this, it's two parts. One is just recently published, where what we see is he has done a really nice study of using high throughput approaches to understand how RNA structure is remodeled in vivo in a non-steady state during embryogenesis after fertilization. And you've seen that the major remodeler of structure is the ribosome. And contrary to what we were expecting at the beginning, that structures would have a major effect on the translation regulation of the embryo, we actually find on the other way around, is that actually translation is the major remodeler of structure in embryogenesis. And then a second part that is unpublished, where we have tried to use a massive parallel approach to try to understand what really drives differences in translation in embryogenesis. So this is one of the core questions of my laboratory, where following in the steps of giants that have really done amazing work trying to identify individual elements across a large number of mRNAs that regulate both translation and mRNA stability, we're trying to use genomic approaches to not only identify many of these elements, but also using machine learning approaches to then integrate how these elements work together to regulate gene expression in embryogenesis. And in particular, today I'll focus on the role of structure. And in this cartoon that JD likes to simplify the idea of using a rubber band to represent the RNA, if you were just looking at the sequence elements in the RNA, you might think, well, you might have all these elements to be active, but of course, when you just put them in the folded structure, you might have some active elements and inactive elements. So we really think it's a fundamental step to understand the function of RNAs to really understand the structure in vivo. So to understand the structure of RNA dynamics during embryogenesis in relationship to the regulation of gene expression, we've used the zebrafish system during a universal transition that happens in all animals after fertilization, where the first steps of development are actually controlled by maternally deposited RNAs. From the work of Joe Richter and others, you can see that these mRNAs really undergo massive changes in polyadenylation and translation regulation. And what you can see is that the very first stages of embryogenesis are actually controlled by these maternally deposited mRNAs up to 3 hours of development. So this provides a unique environment. So up until here, this is all controlled by maternal RNAs and provides a unique environment to really understand what are the post-transcriptional mechanisms that shape gene expression in this context. So Jean-Denis took a brave approach of combining 3 different high-throughput methods, looking at mRNA stability, in polyadenylation, looking at translation changes using ribosome footprinting, and looking at mRNA structure across this non-steady state of development using DMS-seq probing. So in this cartoon, basically the idea of DMS probing, which is adapted from the work of Silver Ruskin and Jonathan Weissman, basically bud the embryos in DMS or use probing in vitro of the RNA, and then through a process where RT will stop in the modified Cs and Ts through DMS, you can then reverse engineer the structure of those RNAs. And we actually, through over 200 million reads of RNAs, we were able to get reliable structure information in over 3,000 mRNAs. And this has just been published in Nature Structural Biology. So, and the idea is that by looking at these non-steady state changes in RNA structure, we can see not only how the structure changes in development and what are the major drivers that shape structure of the RNA, but also we can see how the interplay between translation regulation and RNA structures. We can see how in vivo structure actually predict microRNA activity, and also whether changes in structure can give us regulatory information in the RNA. So from the work of Silver Ruskin and Jonathan Weissman and David Bartel and Guo et al. Have shown that in vivo, many RNAs seem to be highly unfolded. And this is work, just representing the work in yeast from Silver Ruskin. So the identity of the helicases that might unfold these RNAs was unknown. So JD decided to focus on comparing the in vitro RNA with in vivo RNA. And in this, looking for structures that were unfolded or highly folded. So this is a metaplot analysis where basically all the RNAs are aligned together by the start site and the stop site. So you have, this is a region of the 5' ETR, the CDS, and the 3' ETR. And what you can see is that RNAs in vivo tend to be highly folded in the 3' ETR, contrary to what was expected from the yeast work, while in the 5' ETR and the CDS regions, there were decreased structure in vivo when you compare it to in vitro. When we then compare the levels of folding of these RNAs with the levels of translation, what you can see is a clear separation. RNAs that are highly translated have decreased structure in the 5' ETR and the coding sequence, suggesting that this either is a cause or a relationship to translation, while those that were poorly translated were mostly folded, suggesting that indeed translation is a major, either a player on the folding of this RNA or a cause or a consequence. So to distinguish between whether translation and structure, the relationship, whether structure is really driving translation, you can imagine RNAs that have lower structure in the RNA might be highly translated, as we know for many individual cases, or whether translation itself is really remodeling the structure of the RNA. We analyze how the structure changes with translation in embryogenesis, but also block translation initiation as well. If translation is really driving the structure, you would expect that in the absence of translation, now this RNA should have the same structure for highly and lowly translated RNAs. While if the structure is what's driving translation, what you would expect is that if you inhibit translation, these RNAs will still be poorly structured. So this is just one experiment where we inhibit translation initiation through petamin A, which inhibits the translation initiation activity of EIF4A. And what we see is that there are over 30-fold difference in translation efficiency in the wild type compared to petamin A. You see the reduction in translation efficiency. And what you can see is that the poorly translated mRNAs don't change in the level of accessibility when you compare wild type versus petamin A. But the highly translated mRNAs, in the absence of translation, become much more structured, suggesting that indeed translation is really driving the structural changes in these RNAs instead of the other way around. So if translation really shapes the structure, you would expect biological contexts where changes in translation for the same mRNA are driving changes in structure. So there are two well-known events that change the translation efficiency. One is polyadenylation, where many RNAs are initially deposited deadenylated, and through the effect of cytoplasmic polyadenylation signals, they get rapidly polyadenylated, and this causes an increase in translation. And also, upon activation of the genome, after the phase of silent gene expression, we have a massive expression of this microRNA, miR430, that drives translational repression of the RNA. So Jean-Denis looked at the changes in structure between early stages before microRNAs are expressed and late stages once microRNAs are expressed and there is full polyadenylation of these transcripts. So the way we're going to look at these mRNAs is, again, is instead of, we're looking at 5' CDS and 3' regions. And through a different metrics of looking at changes in structure between the two stages, what we'll see is a red label for an increase in structure at the late time point, or a green label or blue label by a decrease in structure at the late time point. And what you can observe, I hope, is that most of the RNAs in this transition undergo a significant decrease in structure. In fact, I hope you can see that there is a stronger decrease in structure in this N versus this N, and that's because Jean-Denis really sorted these transcripts where each line represents a transcript from high translated to low translated. And in fact, if we just look at the extreme of the population and we do a meta-plot of these genes in these two extremes, you can see that highly translated genes really undergo a dramatic decrease of structure at later time points when they have high levels of translation, while mRNAs that undergo translational repression, many of which are microRNA targets, actually elicit an increase of structure throughout the transcript, suggesting that indeed biological contexts that change the levels of translation in embryogenesis can have a significant effect on the structure of folding. So we have seen that there are structural changes and that translation is a major remodeler of RNA structure during these non-steady state situations during embryogenesis. So what can we learn about the regulation? I'm not going to show you data, but there's been extensive amount of work showing that structure can poorly predict the level of microRNA-mediated repression. And what we observe is that we gain a large level of predictive power when we look at the structure in vivo, which is different from the structure in vitro or predictive for microRNA-mediated repression. But what it caught our attention that when we look at structures that change in development, whether they have regulatory activity in embryogenesis. So we looked at a set of dynamic structures that change, and we asked whether they cause mRNA-mediated decay. The first thing that we observe is that these regions that change in structure during embryogenesis actually are enriched for conserved elements, more enriching conserved elements compared to those sequences that don't change in structure. And we also see an enrichment on regulatory activity when we looked at a set of massive parallel reporter assays for mRNA decay. And I'm just going to show you one example looking at the gene nano, which is important for pluripotency and is maternally deposited that is required for the activation of the genome. So this is a genome track on the browser when you can see there is high levels maternally provided early on in development. And as soon as genome activation occurs, you can see that there is a significant decrease in the levels of the mRNA, suggesting that there is mRNA decay. So this is when Jean-Denis looked at the predicted structure based on DMS probing, he found that there was a region that was significantly remodeled based on the structural data. And this region was actually sufficient to cause mRNA-mediated decay, and a deletion of this region, region 2 of 20 nucleotide, was able to abolish mRNA-mediated decay, suggesting that a regulatory element within this dynamic structure is able to confer mRNA-mediated decay in embryogenesis. So just to summarize this part, what I've shown you is that changes in the level of translation have a strong effect at the level of mRNA structure in embryogenesis, in part mediated through mRNA polyadenylation, where highly translated mRNAs are poorly structured, while poorly translated mRNAs are highly structured. We also have observed that changes in the 3' UTR mRNAs tend to be more highly structured in vivo, contrary to the dogma that was drawn from experiments in yeast. And we see that in vivo structure analysis can have a strong power to predict the regulatory activities of microRNA-mediated repression. As I mentioned, 3' UTRs tend to be more structured or untranslated regions that are not unwound by the ribosome. And we see that dynamic structures tend to be, have higher level of regulatory activity and are more conserved than non-dynamic structures in vivo. So while we saw that globally, translation drives structure changes, and we had little evidence for a global effect of structure on translation regulation, there are very clear examples from the literature where the 5' UTR structure has a very strong effect on translation regulation. And we know of multiple regulatory elements, including upstream operating frames, RNA binding sites that can control translation. So Denis decided to develop a method to look at the level of regulatory potential of different regions of the mRNA. And I'm going to show you some results from the 5' UTR. So trying to understand what is the regulatory code that shapes mRNA in translation. So there are challenges to measure translation, and while we have beautiful methods such as ribosome footprinting or polysome profile, there are some difficulties on then identifying the specific variant that might regulate. If you have a ribosome footprint, you don't know which sequence was associated with the 5' UTR, for example, of that sequence. So Denis decided to develop a method where he basically is able to pull down the nascent peptides of translating ribosomes using a flag tag, with the idea is that the purification of these RNAs, you might have higher purification of highly translated mRNAs versus poorly translated mRNAs. He called this method NAPTRA, for nascent peptide-mediated translation ribosome affinity purification. And the idea, and then tested whether this method works. So first was, well, can we inhibit translation initiation and really detect changes in mRNA translation through NAPTRAP? So this is a GFP reporter and a control DS-RED mRNA that is co-injected. You can see how the GFP is downregulated with a morpholino that blocks the AUG and blocks translation initiation. We can see that this can be detected after 24 hours through microscopy, and the NAPTRAP method has a pretty good resolution early on in development to give a snapshot of the level of translation that is going on in this transcript. But can it detect differences in translation from endogenous mechanisms such as microRNA-mediated repression? So he tested two different reporters, one with 3 microRNA target sites versus 3 mutant target sites. And you can see through development, when there is an increase in the levels of microRNA, there is a significant difference in the level of translation between this and this other reporter. So he decided to, so that suggests that indeed this method is able to detect differences in the non-steady state that captures the regulatory activity of UTR elements during embryogenesis. So he decided to apply high-throughput sequencing to this method, where he took over 11,000 inserts of 124 nucleotides, put it in a massive parallel reporter assay that then is in vitro transcribed, injected into the embryo, and then you have 2 parallel purifications, one in the presence of cyclohexamide and one in the presence of puromycin, that will give you the level of background since the peptide is released from the ribosome. In order to, and then apply machine learning to detect the regions that were selected. So, and you can imagine machine learning being a method to really identify what features are predictive of the particular identity of an RNA that is highly or poorly translated. So he initially used these features in collaboration with computational biology Mario Meshish, and found a significant level of predictive power between experimental and prediction through the machine learning using these methods. In the last 2 slides, I just want to show you how we can analyze the features that were selected. So enrichment means enrichment of the feature, and if it's higher than the depletion, means that that feature has a positive effect on translation. So we see enrichment of use in the 5' UTR cause an increase in translation, and this was nicely shown by Wendy Gilbert. We see that an enrichment of Cs has a reduction in translation, and we see how upstream AUGs also has a reduction in translation, as you would expect. So now we have a set of features that clearly AU-rich has an increase in the translation efficiency of the mRNAs, while C have a reduction in the level of translation together with upstream open reading frames, as we would expect, and upstream AUGs. So now we are currently analyzing these features. We are also including structural features in the analysis to see whether we can improve the predictions. And the idea is to now apply this method to detect the different, the regulatory activity and the regulatory code of different sequences in translation in embryogenesis. So finally, I would like to thank everybody in the laboratory for really an outstanding atmosphere. This is how we usually go to work. Jean Denis, who's trying to really kill the structure of RNA in collaboration with Mario and Charles, collaborators, and the initial work was also done in collaboration with Manolis Kelly. Thank you for your attention.
Questions?
Nice talk. I was wondering, the first part where you showed translation is driving the structure. How do you find out whether it's the ribosome actually driving the structure, right? Because you could imagine maybe something about the environment where the mRNA is being translated is driving the structure. Like I can imagine maybe you could look at pause sites or something and see perhaps there is less structure there. You know, maybe that would really tell you that the ribosome is driving
So the main experiments that allows us to see that was the ribosome is the petamine experiment, where we basically block translation initiation, and we see that the structure of the RNA, it's increased only in mRNAs that are highly translated, and it's unchanged in mRNAs that are poorly translated. We also had, so in every event where we saw a change in the level of translation, we saw a unwinding of the structure. So, and then we also looked at whether mRNAs that had very high, let's say globally, mRNAs that tended to have much more structure had a lower level of translation, and we didn't see really a strong correlation there. So it's clear, especially for this audience, that high structure can block translation, but on the global level, we think that, at least in this context, the level of helicases are really reducing the structure in a way that other features have a stronger effect in determining the final translational output of the RNA. At least that's our conclusion at the moment.
Yeah, I just wanted to follow up on that, because I could easily imagine where you would have the correlation you observe, but it would lead you to the opposite conclusion, that structure is really driving translation. So for example, if you have mRNAs with structured 5' UTRs, and that's what's limiting their translation, then of course you need to recruit helicases to get translation, so you'll lose the structure in the 5' UTR at the same time, you know, when translation increases. But it'll still be the structure that's driving the translation, not the other way around.
So what we saw, for example, the mRNAs clearly have a very different level of structure when they have very different level of translation in vivo. In vitro, all those RNAs have exactly the same level of structure. So that suggests that it's something caused in vivo that is really correlated with translation. So, but for us, the key experiment was, we saw that for the same mRNA, when you have a longer poly A tail, you have less structure and you are more highly translated. But the same mRNA, when you have a short poly A tail, you have much higher structure, and we think it's because you're not translated.
But I could argue that with a long poly A tail, you're forming the closed loop, your helicases are working better, so you're removing inhibitory structure from the 5' UTR, and that's what's increasing translation.
Yeah. Yes, That's possible, but the second experiment was when we used the PETamine A, we saw that when we block translation initiation, we see that now all those RNAs gain, that had very little structure, gain structure. So that suggests that it's really when we remove the ribosome, you reduce the structure. However, when we use cyclohexamide, those RNAs remain open, meaning that when you lock the ribosome, those RNAs remain unwound. Maybe I'm missing something, but I'm more than happy to discuss more.
I have a slightly provocative question, and that is whether asking the question whether structure is controlling translation or translation driving structure, whether that is sort of too coarse, too rough a way of looking at things, particularly when you sort of bin it into multi-hundred or thousand nucleotide bins, such as the open reading frame, the 3' UTR. 5' UTRs are usually a bit shorter, but they're still relatively large. And regulatory elements, particularly where they are binding sites for RNA binding proteins, tend to be relatively small, at least can be quite small. Yeah. And then such effects could very easily be diluted out by the rest of the nucleotides in your bin.
Yes, I mean, so I understand that, when I talk to Kevin Wicks, he likes to look at the individual mRNA, the individual nucleotides. In our case, the signal of translation and so correlation between translation and structure is really strong for highly translated mRNAs. But when we remove translation out of the picture, all those mRNAs gain structure. So that's what, I don't have any doubt that strong structures have a repressive effect. But at the level of the embryo, we see that in this dynamic system, that changes in translation are really driving the changes in structure for the same mRNA.
But I'm not doubting what you're seeing. And if you have the iron of the ribosome going over it, it's I guess what one would expect. I'm not talking about structures that per se are inhibitory. I'm talking about structures that are typically weak structures that can be recognized by regulatory proteins. But we can do that at the bar.
And I think the next step is to look at the individual structures in all these mRNAs.
Okay, I hate to curtail a good discussion, but we have to let all the speakers have some time. Thank you again, Antonio, for a very great start.